Hello, you're already here. Thank you for opening the room. How are you today?
Oh, great. Thank you for inviting me. Can you hear me now, Katerina?
Yes, I can
Good.
hear
That's
you
great.
well.
Yeah, because this is my first time to use this app, so I'm not quite sure I
Yeah.
use it correctly. But thank you for arrange everything for me.
Oh, of course. If you could do me one favor,
Yeah.
could you make me moderator so I can put the link for your presentation and so on? So how you do it is you click on my profile picture,
Mm-hmm.
and then there's an option on the bottom, make moderator or something with moderator.
Yeah, I think I just did.
Perfect. Thank you so much. Yeah.
Mm-hmm.
Sorry that I came later than you.
No, don't be.
Okay. Hi, Jamie. How are you? Please meet Jamie. Jamie, please meet Dr. Huang.
Hi,
Hello,
Jamie. This
Doctor.
is
Very, very
Zhenqiu.
nice to meet you.
Nice to meet you here.
Thank you. Thank you for coming today.
My pleasure.
Oh yes, please. How do I pronounce your first name? I don't want to say it wrong.
So my first name is Zhenqiu. I think it's very challenging to pronounce it, but doesn't matter.
Zhenqiu.
Yeah,
Zhenqiu. Okay, thank
that's
you.
perfect. Mm-hmm.
Yeah, my name is also hard to pronounce, so I
Oh,
know
yeah,
the
please.
challenge.
Yeah, is
Oh, don't
could
worry
you?
about my name today. It's just other people. Yeah, let me add the link of your presentation. And thank you for sending it. Great. And we'll wait around 4 more minutes to start until
Sure.
it's perfect. How's your day today? Finally, a little bit
Good.
of sun.
Yeah, how are you?
I'm good too, thank you.
Mm-hmm.
This week is a little bit less busy than the last week, so I'm
Oh,
glad.
okay, that's good.
Great. So yeah, the link is there. The topics are there. Perfect. Okay.
Thank
We'll
you.
wait a few more minutes. Good. So yeah, it's the first time you're here on Clubhouse. So thank you so much for doing that, for joining Clubhouse for us and going through that. We really appreciate it.
Thank you.
I hope
Yeah,
it was not too much trouble.
no, not at all. It's my
Yeah,
pleasure.
it's there's a lot of you can on Clubhouse find rooms about any topic that you can imagine.
Oh,
so,
okay.
so this started more during the time of COVID when people were stuck at home. It became very popular quite fast. So
Okay, I see.
yeah.
How do you organize this? Are most of you from NYU or?
No, so actually I joined Clubhouse in the beginning when Clubhouse started. You needed an invitation. Hi, Cecilia.
Hello.
I think there were mostly like some famous people here and media people. So you needed an invitation. But my husband was working at that time at Vice Media. So he invited me, but I didn't use it too much. But then, yeah, being more stuck at home, I listened to different rooms where science topics were discussed and so on. So
Oh, I
then
see.
people asked me to talk about my work, and then I thought, oh, it would be interesting, maybe an interesting platform to talk more about science. So I started inviting people, and yeah, sometimes there are really great conversations going on. And yeah, then I created this club because people were interested to listening
I
to
see.
science. Yeah.
That's great.
I mean, you can find also like crazy rooms here on Club, like everywhere on social media, but this is kind of counterbalancing that a little bit.
Sure.
So
Thank
yeah,
you for organizing this.
and so we have members from all around the world. So we
I
have
see.
a different time zone, so that's why this room is recorded. So people from other time zones where it's in the middle of the night, or for example, some of our members, or quite a lot, are from Asia, so it's in the middle of the night, or when it's getting later, they listen to it afterwards. So that's why it's recorded. So
I see.
it's kind of a way to make an interactive podcast. So in the meantime, while we're recording, people can ask questions.
Sure. Yeah, that's really a good idea.
Thank you so much for giving this platform a try, Doctor. I've only been in this the clubhouse briefly, and I've been amazed at the ingenuity people have had with the Katerina says, all the different rooms. That's how I found myself to Science Society, because Katerina's amazing at getting incredible guests like yourself. Because I wouldn't be in a position to actually get to ask any questions to people like yourself. So it's quite exciting.
Yes.
Okay, I think we can start. So welcome everyone to the Science Society, and we are very honored to have here today Dr. Huang, who will talk about this paper that was recently published, and that's very, very interesting, and I think also very important work. It's cancer related, as you can see in the title, but let me first introduce you a little bit to Dr. Huang. He did his education at Sun Yat-sen University. He did there his master's degree in microbiology and a general master's degree. And then he did his PhD in cellular and molecular biology at the Czech Academy of Science Institute of Parasitology Biology Center of the Czech Academy of Sciences. And then he did his postdoctoral research at the Arizona State University. And currently he is at the Albert Einstein College of Medicine in New York City. And there he studies genetics via single-cell technology, which brought us now here to this very interesting research. So welcome and thank you so much. And if it's okay, Jamie would like to ask you like a more general question in the beginning before you start your presentation, if that's
Sure.
okay with you.
Yeah,
Perfect.
thank you for the introduction, Katerina.
Yeah, thank you very much, Doctor. One of the things that we actually found really interesting here is to find out what actually got you started on your path to science and specifically into the study that you found yourself in. So could you tell us that where did you begin? Did you always have this goal in mind, or did you have another dream when you were, younger and coming up through school? What fascinated you about this?
Yeah, so thanks for asking. So since I was in my teenage, of course I did hear, and even it had happened to us that some of my relatives, they got like the cancer, but that time I believe the therapeutics was even not as advanced as nowadays, and I was as a teenager already started to think about what I can do, if in the future I have the chance, will I be able to join the scientific team to develop some method to either diagnose the cancer at the early stage? I mean, the whole idea was not so mature back then, but slowly it did influence me to choose the subject I want to begin with. Yeah.
But it actually came from a desire just to make things better for people like your family. That's incredible.
Yeah, it is. But later on, of course, there's always ups and downs during the pursuit of this kind of dream. Yeah,
Yeah.
because to be honest, at the very beginning, we started with basic science, because in the college where I studied, it's not that each section was so closely related to cancer study or genetic study. So, as Katerina just introduced, my path was like very winding. I tried different subjects, and in the end, yeah, I was really lucky that I can study what I really like.
That's wonderful to hear. Um, that's great. So you had to study many other things just to get along this path then, because you had to know so many things about so many other things. Is that right?
Yes, it's true that uh, while I'm in the path to reach where I am now, yeah, well, I did learn and acquire a lot of knowledge from different either biology field, uh, microbiology, parasitology. Yeah, they are very diverse, but they did build up my basic knowledge for where I am today.
That sounds remarkable. This is actually why we've been particularly enjoying this club because we found when we get multiple speakers from different areas, we can have all the ideas coming together. So it sounds like you were already doing this yourself. So thank you very much for sharing that. If you'd like to just begin your presentation, we are all ears.
Thank you.
Is that okay,
So,
Katerina?
Yep, thank you so
Yeah.
much.
Okay, thank you.
so I will start to present, okay?
Yes, thank you.
Absolutely.
Okay, so it is my great pleasure to talk about the project I've been working over the last 2-3 years. And the topic is single-cell analysis of somatic mutations in human bronchial epithelial cells in relation to aging and smoking. If you come to the second slide from the link, you may notice that in this graph, it is demonstrated lung cancer is by far the leading cause of cancer deaths among both men and women, making up almost 25% of all cancer deaths each year. More people die of lung cancer than any other type of cancer, and it may be the number of people died from lung cancer can be the sum of colon cancer, breast cancer, and prostate cancer combined. So coming to the next slide, in this graph plot, it shows that cigarette smoking is the number one risk factor for lung cancer. So in the United States, cigarette smoking is linked to about 80 to 90% of lung cancer deaths, and 10 to 20% of lifelong smokers develop cancer, which is a lot if you think about the huge number of smokers. A lung cancer risk in case control studies show the relations between lung cancer risk and smoking dose. So the smoking dose here refers or demonstrated at the bottom of this slide is calculated by the years of smoking and the average packs of cigarette per day. So I will give you an example. So if a smoker, he smoked 20 years and each day he smoked just one pack, then his pack years is 20. But if he smoked 2 packs per day for 20 years, then his pack years is 40. So this is a kind of indicator of his lifelong smoking dose. And we did find that the higher the smoking dose, the higher risk of the lung cancer. But it is another reality that most of the lifelong smokers or heavy smokers, they don't get or develop lung cancers. So in the fourth slide, here it showed apparently that when you do not smoke, of course, the cancer risk is minor. When you smoke continuously, the cancer risk increases significantly at advanced ages. We can see here at the age of 75, yeah, it is with the highest risk of lung cancer with those continued smokers. So in the fifth slide, we are asking that how may tobacco smoke cause lung cancer? Because we all know that those some heavy smokers did develop lung cancer, but how did it happen? There must be a biological cause of it, right? So it is generally in this schematic illustration, it demonstrated the basic mechanism or potential mechanism how smoking can cause lung cancer. It is because the carcinogenic chemicals in the tobacco smoke, including the polycyclic aromatic hydrocarbons and other carcinogens, they induce DNA damage and increase the erroneous DNA repair and replication, which generates DNA mutations. During the continuous smoke exposure, mutations accumulated in cancer driver genes, including proto-oncogenes and tumor suppressor genes, and cells were under the selection for growth advantage and tissue invasion, and eventually evolved into cancer. So with the critical role of mutations in causing cancer, the assumption of how smoke-induced cancer has not been proven, as nobody was able to show or measure mutations in normal cells exposed to tobacco smoke. So in the sixth slide, here generally showing you the clonal expansion of initiating cell during the cancer development. So it is much easier to detect somatic mutations in tumor cells. This is because most tumor could be treated as a single clone. Basically, they just expanded from the initiating cell at the very left. Therefore, when it clonally expanded into a tumor, all the mutations in common of all the cells may provide information of the original mutation level or original mutation pattern in the initiating cells. However, by now, little is known about the normal cell, because as we showed in the sixth slide, with the tumor, we may only get the mutation frequency of the initiating cell. And that is the moment when initiating cell transformed into the cancer cell. With this concept in 2016, Alexandra used the tumor tissue, which were clones from initiating cells, as a surrogate for the initiating cell to study somatic mutations. Yes, exactly. They just used the tumor as a surrogate to find the sharing mutations between each cell so that they have the information of the initiating cell's somatic mutation. But they did not really study the normal cell, which means before transformation into the cancer cell. And here our hypothesis is that somatic mutational burden increases with age in normal lung cells of never smokers. That means the mutation may accumulate with age growth. And this kind of mutation will be elevated by smoking and dependent on smoking dose in the normal lung cells. Yeah, we brought up our hypothesis on slide 8. So in slide 9, here is to demonstrate the method or the strategy to analyze the somatic mutation level in the normal cell. On the left side, it shows that shows how the mutation developed when it is from the zygote. We can see here that during the early embryo stage, the red dot representing the somatic mutations. It will only be found later among a few different cells. That is where them arising from. But most of the somatic or most cells, they are not sharing the same somatic mutations. That's why to find out the mutation frequency of each cell, we need to use the single cell method. On the right side, it is show the principle of how the single cell method work. So basically, our lab developed whole genome amplification method on single cell to find somatic mutations. The true mutations could be identified in this method because they always linked to the germline SMP. SMP representing the single nucleotide polymorphism, whereas the errors during the amplification and sequence are easily randomly present in all sequencing reads. But by link to the SMP, we'll be able to distinguish the true somatic mutation and errors. So with this method, on the slide 10, in previous studies from our lab, mutation frequency was found positively correlates with age in human B lymphocyte, which is at the left, and hepatocytes at the right. And in slide 11, these are our specific aims. M1, we want to optimize a pipeline for the single-cell somatic mutation measurement in human lung. M2, analyze the somatic mutations of normal lung cells in relation to age among never smokers. M3, analyze somatic mutations of normal lung cells in relation to smoking status. Here, so on the slide 12, here is to show the accomplished M1 with all subjects, that is, harvesting the proximal bronchial basal cell from the endobronchial biopsy, which was performed during bronchoscopy in the bronchus at the bifurcation point of the lung. The bronchial brush was employed to brush off proximal bronchial basal cells, which are candidate progenitor cells for squamous cell carcinoma. The brush with all cells were then cultured in a selective media for short term, and the normal bronchial epithelium were screened and kept as frozen pellets in the repository for logistic effectiveness. These two steps, step 1 and 2, were accomplished by our collaborator, Dr. Simon Spivak's lab. Our lab continued with nuclei isolation from these frozen cell pellets and collected single nuclei for each subject. With single nuclei, we performed whole genome amplification, library preparation, and whole genome sequencing. The sequencing data were analyzed to identify somatic mutations, which include SMV, abbreviation, that is abbreviation for single nucleotide variations, and indels, abbreviated for short insertion and deletions. This pipeline with nuclear isolation step enable the analysis of samples that could not be harvested and processed in one day. The same pipeline could also be adapted for live cells. So in slide 13, I would like to introduce you the human subjects in this research. For this, as we already set our aims, so here 13 subjects were recruited. Among them, 31 were under the endobronchioscopy for the suspect mostly of lung cancer. It was in this process when the lung cells were harvested. There were 12 never smokers with bronchial biopsy. Their age were between 25 to 86. Additionally, we commercially obtained the same primary cell from 2 teenagers. That is for the M2 to find the relations between mutation frequency and age. The smoker group include 19 subjects. The cumulative smoking deaths were indicated by pack years, with cancer and cessation status listed accordingly. So here is the result. We measured the mutation. On the slide 14. Here is the result plot. We measured the mutation frequency in 51 single nuclei from 14 never smokers. So each dot in this plot represents a single nucleus. We found the SMV frequency, that is the mutation frequency, as indicated by a y-axis, increases with age of donors, indicated by the x-axis. At the scale of SMV per megabase, we found the cell of the highest mutation, which is almost at the top of the plot. It's very close to the mutation frequency in lung tumor of a never smoker in the previous study we referred. It is tempting to suggest that this cell may be the initiating normal cell of cancer development. We calculated the median SMV frequency of each never smoker, represented by each dot in the plot. The orange bar indicated the minimum and maximum mutation frequency of 3 to 5 single nuclei measured per subject. We found the SMV frequency increased with the age of donor, which corresponds to a rate of 28 mutations per cell per year. Similarly, the frequency of indels, that is small insertion and deletion, also increased with age among never smokers. However, the rate is much lower. The rate is around 2 indels per cell per year. On the slide 17, to find smoking status effect on the somatic mutation, we plot smokers against age of donors along with never smokers, represented by red and orange dots respectively. So the red dots indicating the smoker's mutation frequency, while the brown dot indicated the never smoker's mutation frequency. Clearly, we can see that the mutation frequency among the smokers are significantly higher than never smokers when they were all plotted against age. In other words, since smokers' mutation frequency elevated compared to never smokers. Indeed, when we compare the rate of mutation frequency by year, we notice that in smokers it is 91 SNV per cell per year, whereas the never smokers only 28. Overall, then on slide 18, we also try to find the indel frequency among both smokers and never smokers. Overall, indel frequency among both smokers and non-smokers increased with age. It was estimated that rate of indel among smokers was twice of that of in never smokers. On slide 19, to gain better insight into the relationship between smoking and the mutation burden, we analyzed mutation frequency in the lung cell as a function of smoking dose. Among the data points, there's one outlier that is very obvious, indicated by the number 1320 at the left upper part of this whole plot. This is a light smoker at the age of 81 who was also a cancer patient, and it contained a very high number of mutations in 4 cells sequenced. We then analyzed the mutation frequency as a function of pack years with or without this outlier, 1320. So when we analyze without the outlier, mutation frequency positively correlated with smoking dose up to 23 pack years. After that, surprisingly, a level off was observed among smokers with heavier smoking. The switch point was found statistically significant by our likelihood ratio test with piecewise linear model and B-spline model. When the outlier subject was included, the nonlinear increase of mutation frequency was essentially the same. So irrespective, the result clearly showed that the mutation frequency in lung cell of heavy smoker was not higher than the moderate smokers. The indel frequency was found not, on the slide 20, the indel frequency was found not dependent on pack years either. On slide 21, we tried to plot the mutation frequency with the lung cancer probability, with the y-axis at the right indicating the probability. So in this graph, we can see that it seems while the mutation frequency did not go up after 23 years, the lung cancer probability among the smokers, they are also not showing a linear increase. So there's indeed some evidence that lung cancer risk may go down slightly at very high smoking dose or high pack years. Our show that mutation frequency do level at higher smoking doses. The cancer risk has been calculated over populations, and we analyze individuals. The fact that 80% of heavy smokers are not with lung cancer, as we previously introduced, and our data here offer a possible explanation, that is, individuals' cancer risk may depend on genome sequencing integrity, which may be different from individual to individual. Thus far, no one could measure that in normal cell. So to analyze, on slide 22, to analyze the mutagenesis from all subjects, we applied a computational method developed by Alexandra based on cancer tissue, which are essentially clones of cancer cells. A database called COSMIC demonstrates signatures extracted from cancer tissue and linked them to mutagens such as tobacco smoke, aging, etc. The SNV mutation spectrum of smoker and never smoker at left showed relative contribution of each 96 mutation type, with the 6 base substitution type indicated at the top. The right histogram showed extracted mutation signature with their relative contributions. Among them, aging signature were dominant in both groups, whereas smoking signature was evidently found only in smoker. In slide 23, similarly, we analyzed the signatures of indel. The spectrum of indel at left, with this top indicating insertion, deletion at different lengths by color. In the histogram of the major indel signature at the right, the smoking signature in green was evidently higher among smokers, where other signatures shown were either novel or have not been linked to any clinical mutagens in the database. As mutations, so in slide 24, as mutations in cancer driver genes were critical for the transformation during cancer evolution, we searched among all 134 cells measured from these 33 subjects, and found 21 mutations from 12 lung cancer driver genes out of all 100,000 observed somatic mutations, with gene, number, name, and type of mutations indicated at the left diagram. Driver mutations were mostly from smokers. Further, we asked whether more driver mutations found in smokers were due to the overall higher mutation frequency, as we previously mentioned. To answer this question, we compared mutation frequency in driver gene versus non-driver gene. In the plot at the right, the red diamonds indicate the mutation frequency in lung cancer driver genes with normalization of covered genome size, while the boxes indicating median and the interquartile range of frequency of mutations from lung cancer driver genes, up to 200 times repeats. Yes, that means due to the overall higher mutation frequency in smokers, mutation in lung cancer and the pan-cancer driver genes were more frequent in individuals with a smoking history. On the slide 25, to evaluate the possible effects of genetic background on mutagenesis in lung cells, we tested all studied subjects for the presence of germline variants, that is, the genetic backgrounds previously associated with the solid cancer or listed in the ClinVar database, that is a clinical database with the pathogenic variants. So out of the 33 subjects, a total of 17 were carrying at least 1 or 6 identified genetic variants associated with risk of solid cancer. Among these 17 subjects, there were 12 the lung cancer cases and 1 other cancer cases as indicated by red box. So this enrichment of risk of cancer risk variant in cancer cases was statistically significant. On slide 26, I would like to summarize our whole study. We analyzed somatic mutations in 133 normal lung cells from 33 subjects. The somatic mutation frequency increases with age in normal lung cells of never smokers. The somatic mutation frequency is elevated by smoking, but level off in heavy smokers. Signatures related to tobacco smoking are found with significant contribution only in smokers. Lung cancer driver gene mutations were found, but not significantly different from mutation frequency in other genes. Cancer-related germline variants enriched in cancer cases. So in slide 27, here are our future direction. We would like to measure other somatic mutations, for example, genome structure variation and copy number variations. Also, we would like to analyze the somatic mutation landscape of blood and buccal cells and compare with lung cells of the same subject. Further, we would like to explore the germline mutations in genome maintenance genes as risk factors for high somatic mutation burdens. With that, I would like to thank my mentor Yang and all lab colleagues. Also thanks to our collaborator Dr. Simon Spivak and his lab members, as well as Kenny Ye for all the statistic analysis. And I'm happy to answer any questions. Thank you so much.
Yeah, thank you so much for this very interesting presentation. It was really great how you explained the data. And the first thought that I had when I first just read, the abstract of your paper was, we had the guest speaker here before showing Detlef Weigel. He works in plants, but he showed basically that actually in plants mutation don't seem to be completely random after all, and that they detect over generations less mutations in very important gene sites that are important for survival. So I thought that maybe your work would point it would basically maybe support a little bit that this is also happening in animals, but you say that the location of the mutations, you didn't detect a pattern that's important regions of the gene for different function, maybe even protective functions of gene repair. They happen everywhere randomly. That's how you see it, right?
Yes. So I did know that. I think the research you referred to of the non-random mutations in plants, even though I did not thoroughly read that paper. In our case, I think when mutation occur, it is randomly distributed in the genome, but at the same time, there's a selection of mutations. So initially, if the mutation occurred for example, in some vital genes, but the cell may not be able to survive. So this kind of mutation, it happened, but we will not be able to capture it, right? So this is the selection process in the human body. Whereas there are some mutations, it could be selfish mutations, which can give the cell advantage of growth. Therefore, it will proliferate much at a greater rate than other cells. So this kind of cell may possess the potential to transfer into the cancer cell, because it may outrun other cells. So this is kind of selection in our body. In the end, we are just measure the terminal mutations levels of the cell, which we can harvest it.
Yeah, that's really interesting. Jamie, you had a question?
Yeah, I was going to thank you for the talk, and it was fascinating when you were talking about the levels of mutations. When you said like never smokers were what 28 mutations, and there was smokers being 91. I was wondering when you said about lung cancer being so much more prevalent than loads of other types of cancers. Did you know why lung cancer is like particularly bad for this, or the lungs are particularly bad for these kind of mutations and things?
Yeah, so the slide I'm talking about, I guess, about the lung cancer is the leading cause of all cancer mortality. That is on slide 2, right? So here, the prevalence of cancer, the prevalence of lung cancer may not be necessarily the leading one, because as far as I know, the skin cancer is the most common cancer, or the with the highest occurrence in the world. But the lung cancer is the leading cause of mortality. That's because either it is based on the survival rate, I believe, of the clinic data. Why it is like that? Or sorry, am I asking the question am I answering the question you're asking?
Oh no, yeah, no. Please continue, because like I said, I'm fascinated just with you saying that skin cancer is like higher. But yeah, no, please continue. Yeah.
So the mortality rate, I think, it is. I did know that because among even the lung cancer, there are many different types. One of them is called squamous carcinoma, which usually, when it is diagnosed, it is already at a very late stage and very difficult for effective therapeutics to apply. That's why it had very low survival rate, as far as I know.
I actually had squamous cell carcinoma, and one of the things that was surprising about the statistics, because I kind of jumped in being a biophysicist, I was like, I want to understand this problem. And one was most people, if they catch this kind of cancer late, that is stage 3 or 4, there's less than 20%, something like that, survivability. Whereas if you get it treated earlier, there's a high survivability because the chemotherapies are quite effective. I think one of them is called BYL719. That's the chemotherapy that I took for that specific cancer, and it was some sort of PIK mutation. Yeah, and in particular for the stage 1, stage 2 cancers, most of them are operative, meaning you can just do surgery and physically remove that tissue. And so that's kind of how it breaks down, at least from what I understand. So it's high survivability if caught early, and somewhat treatable for most people. So
yeah, thank you for sharing.
Do you also think that in the current climate with like really bad air all round about us and things like that, that there's like a sort of chance of triggering more mutations in the lungs? Like, because I mean, if we're breathing in bad air for pollution and things like that, then it's essentially like a sort of type of bad smoking, but just very, very, mildly over a long period of time. Have you
Yeah,
seen anything about that?
so for the about the risk factor of lung cancer, apart from the cigarette smoking, we also noticed that even though I only work on the genetics and the molecular mechanism at a genetic level, but I did read that so especially in the early 80s or 90s, it is found that in Asian country, which especially among women who I think cook with the old stock, where there was a lot of smoke from the burning material. They also have high risk of lung cancer occurrence. Yeah, and of course nowadays because of the pollution, this is an add up risk factor for lung cancer occurrence also. But all this because in our study or in our further study, we are trying to find the genetic background of at the individual level, because it seems each individual or when it has come to the population level, not everyone show the same resistance to the cigarette smoking caused mutation or even cancer. And that may help us to find out why someone is more susceptible while the others less. And we hope we can find the for example the genetic background that may determine all this, and we may use this information for the future application, translational application.
So trying to
Have you
find the
looked at?
people. Oh,
Sorry, go ahead,
go,
Jamie.
go. No, please.
Have you tried looking at, like, one of the things that I know Craig Venter looks at? So from John Craig Venter, the guy who sequenced the human genome on the private industry side, he talked about, like, one of the ways that they partition their data was to look at people who didn't make it into old age. So separate the population into people who, half of who died by 50 and then the other half who died after 50. And so you get this kind of bimodal distribution. And for example, like, not to make it sound sad, but some people have certain genes that are not found in older populations. So we know that those people just don't make it to that age, and perhaps differentiating subpopulations like that. So I guess my question is, have you started doing that analysis? Because it wasn't clear from the way that you phrased it whether that was the question that you were going to be answering next.
So we are not directly to your question. We are not doing the population or the genetics at the population level. We are still trying to. We are more like trying to analyze the mutational level to track back what could those people who are more resistant, and if we can find those people are more resistant, we'll be able to survey their genome and to find because the whole genome is really a very big pool for us to find out. But if we can locate those people who are more resistant, we will be able to maybe do it more precisely. But at the population level, yeah, it is. I think the whole genome is really big. I think the way the research you describe may still provide us still zillions of data. Then we still need to screen which one or how they coordinate it with each other, so that it can determine someone's lifespan in the end.
Oh, okay. Yeah, so you're going very like computational model, I guess, is most of
Uh,
the kind of.
just so it's in the presentation, I know you came a little bit later, but this is looking at single cell somatic mutation measurements in the human lung from specific people. And then you have never smoker 12 subjects and smoker 19 subjects. So um, if that helps.
Yeah, I guess I was asking like how was the study like what is it called, pre-registered or whatever? How was the population selected? Um, I guess that's probably just an elective thing, like
Okay.
in a clinical trial, right? So I signed up for a clinical trial, so I guess that's how that was. But in terms of starting to look at a problem like this, you can look at the molecular variations, but how did that question get simplified to that cell, I guess?
Yeah. So all these subjects, they they were doing the biopsy because they are in a suspect mostly of lung cancer. Yeah. Uh, or structural abnormality. So that's how we got all these subjects, and we are more like a quantity or analyze uh, the terminal what's that? The terminal end point, which is the mutation, because uh, they are from the DNA damage and to repair and all the process and selection. In the end, it was just demonstrated on the cells mutation level, and that is something we are measuring on. So we are trying to use that. We're trying to analyze the mutation level to evaluate the cancer risk. That is basically what we're doing.
Okay, so I guess were the biopsy samples sequenced beforehand, or like were like I'm curious about the distribution because the data set seems small, like it's not in the thousands, right? So I'm
Yeah.
curious like what was the power of the study like statistically, because that seems like an impressive extrapolation from such a small set. So
Mm. So as we are doing the whole genome sequencing, firstly, I need to stress that it is very expensive to have the whole genome sequencing data out of 134 cells. So in this our current study, there are only 134 cells from 33 subjects, and we yeah, it seems at this moment the subjects number is not high compared to the population study. As I pointed out, for the cancer risk study, usually they use the population to make the statistic model. But in our number is lower, but we are still our results are all based on the statistic significant analysis, and that's why we may want to develop uh, the alternative for survey the mutation frequency in blood or the buccal, so that we'll be able to uh, in to uh, to have a bigger pool of the subjects. Yeah, that may help us to find more information for screening in more subjects cancer risk by this kind of method.
This is fascinating. And so, are you hoping then? Sorry, please correct me if I'm misunderstanding. But you're looking at the genome of people, and you're trying to find where certain people are more resistant, and then you're trying to go, why are they more resistant, or something like that, so that you can perhaps find a way to give this resistance or to other people. Is that correct?
Yeah, so I think a very easy example for us to start with is all of us may heard BRCA1, right? BRCA1 genes deficiency. And the idea is maybe, and it is just a small nucleotide polymorphism. So it is just a single nucleotide mutation may affect the function of certain gene. Therefore, when it accumulates, the effects accumulate, it leads to higher cancer risk or lower cancer risk. But so the idea is like what you just mentioned. If we can find someone or some people, they are more resistant to mutation induced by smoking, we will try to locate their polymorphism or single nucleotide polymorphism, so that we may use the CRISPR technique to analyze the effects of this SMP.
Oh, by then turning it on and turning it off, I guess, and
Yes.
kind of letting. Okay. Yeah, so I guess there was enough consistent damage from smoking that there was enough similarity in the cells, or I guess that's what the power of the statistics kind of played out, that it didn't matter.
Sorry, can you repeat your question again?
So I'm just asking about the consistency of the damage to the genome from smoking. Was there what was I'm looking at the slides and I don't know if it's actually presented in the slides, but I'd be curious about the distribution of the mutations, like what was the clustering? Because there's some mention in here, but
so if we
like
look
somatic mutations or that slide.
If we look at the slide 17, you may find, well, on the slide 17, the red dots are the mutation frequency of smokers, and apparently they are significantly higher than the never smokers of the same age, right? So mutation frequency, yeah, this is like the mutation frequency at the general genome level. Then further, if we look at the slide 24, at the left side here is about mutation found in the cancer driver genes, which are the driving force of cancer development or cancer cell proliferation. And most of these mutations are found in most of these mutations are found among smokers, but it is not because they are selectively occurred in this gene, but because of the overall high level of mutations we showed previously among smokers. Yeah. So
okay. So there's like an average. So there's an average drift of the amount of mutations kind of moving up from. Okay. Yeah, this is exactly what I was asking. Sorry, I'm not sure if you went over that slide, but thank you for going over it now.
Yeah, my pleasure.
And I'm wondering as well, like with this research, are you hoping that if you can discover like this resilience or be able to give it, is this meant to help with prevention, or are you hoping to find something that would reverse these kind of mutations and problems for people who have it already, or are you looking at this as likely going to be people could maybe be given, like, I don't know, like a lung cancer vaccination or something to give resilience?
I think here our study is more like cross-sectional study, so it is at one moment. Like I said, if we can develop or validate the mutation analysis from the blood or from the buccal, because that would help us easier to track the mutation development in the normal cell, and hopefully we will be able to find like more targets for the chemotherapy or for the targeting by the antibody. But at this moment, with our current study, I don't think we can realize or make it what you suggested come in anytime soon.
Right, right. It's something we'll look forward to. Thank you very much, Doctor. That's fascinating.
Yeah, I
Thank
just
you.
wanted to comment that I think this is a great model for studying resilience against cancer because smoking, like to basically to have a real life model and framework to work with in humans where such a impactful factor is taking place, because using other pollutants, it varies so much, right? If you live in the city, if you live in the suburbs, if you live out on a farm, there are different pollutants affecting you. So I think you found a really wonderful model and started creating this framework to look into resilience against cancer and comparing it to aging, because aging is still the biggest risk factor, as we see also very nicely in your slide. So I want you to compliment on this and starting this study, and it's a lot of data and a lot of work. So it's really wonderful, and I hope I have a lot of, personally and also from research perspective, I have a lot of hope for changing the perspective to asking resilience questions, so that we can go forward and in the future maybe help people better. So thank you. This is really wonderful. And I
Yeah,
wanted to
thank
check if you have, if anyone has some last questions, because we
Yes, me.
our, yeah, go ahead.
Yeah, so you mentioned like I guess monitoring or more consistent monitoring that would allow you to keep track of the changes. What kind of technologies could you envision or would you want to see? Like, and I guess one thing to note is that I'm an entrepreneur who has a biotech company that makes a type of sensor that makes certain sort of measurements on biomolecules. What would be kind of the dream device if you could envision one that would allow you to perhaps make some leeway on this kind of problem?
if you ask me to envision, I think here something I think in the future we may be uh, check more intensely. First, of course, is sequencing. If we can to do more high throughput, either whole genome or single cell, uh, either single cell or box sequencing, we'll be able to track the development at the genetic level or molecular level. Uh, Secondly, I think what now people trying to do is trying to correlate the imaging signature with the genetic signature. And if that can be developed so that in the future the high resolution imaging device may help us to capture or diagnose the cancer at the its earliest stage, that can also help to take more or early preventions. Thirdly, I think probably still it is back to it is about sequencing, but it it is to targeting or it is to capture something like cell-free DNA or I don't know stability of RNA in the blood, so that may also help us to get the information of carcinogenesis at this early stage. That is my viewpoint.
Yeah, that's awesome. Yeah, our team is looking at I guess metabolic byproducts such as double-stranded RNA and other things like that that are early indicators of changes in the system, and we're trying to see with some I guess agriculture and then perhaps soon with some animal models, we'll be able to get some more testing done and see if we can actually make that correlation, because it seems to be like an open problem, and deep learning or the field of automation or machine learning seems to have quite a lot of fruits to bear. And this problem just seems like it should work, although I think it's often a measure of the technology of the time. So it's an interesting time now to see, because most of the signal generator technology that's used in cellular networks lends itself to biotechnology sensor development. And so the costs have really come down quite a lot in the last few years. So it's definitely an exciting time. So thank you for entertaining that question. And it would be really awesome to speak with you a little bit about that, if possible, about
Yeah,
kind
of
of what
course.
kind of capabilities you would want. Because we enjoy not only hearing about the kind of work that you're doing, but also the things that you need in the laboratory, and maybe someone that we know or whatnot
Yeah, actually,
might be
just
working
to
on
let you
something like
know
that.
that, I'm sorry, our lab, I think the single cell whole genome sequencing is just only one technique developed in our lab that I'm talking about today, but we are also trying to develop in different methods. Recently, I think our lab just published another paper called single molecular method of sequencing that is published on Science Advance. If you are interested, you may, because it is based on the box sequencing. It may also, instead of isolate the single cell, it may, by extracting the genomic DNA from either tissue or from blood, or even it has been tested with cell-free DNA to quantitate the mutation frequency, and we were hoping to leverage this method for the further use of disease risk evaluation.
That's so interesting. Maybe we can have another room, maybe in June or so, if you would be willing to come back and talk about this
Yeah,
method. Sounds like
sure. Yeah, I mean
important.
that method is many. I was a co-author of that. I mean, co-author of that technique, but the first author, I can talk with my colleague. Yeah, probably he will be the best person to introduce the method.
That
Yeah,
would be
yeah. Thank
fascinating.
you. That's
Thank you very much,
beautiful.
Doctor. Yeah, thank you.
You would have time in June, sometime in June. That would be great. Would be really interesting to have a follow up room basically and talk about this technology
Mm-hmm.
from the lab. So,
Sure,
do you have time for one more question? I saw that five
sure.
joined. Yeah, thank you. Five.
Oh, thank you. So very interesting work. Thanks for sharing. I just went through the slides because I did join a bit later actually. I joined maybe a couple minutes ago. But the slides were very helpful to catch up and sort of get a really good idea about what you did there. I was wondering if you did any subgroup analysis in terms of the type of lung cancer, because we know from as a doctor essentially, we know patients that smoke tend to get a type of non-small cell lung cancer, specifically named squamous cell carcinoma,
Yeah,
as opposed to, for example, most non-smokers getting the another type that most of the time,
adenoma.
obviously, the adenocarcinoma, exactly. So I was wondering if you had any insight into into, any subgroups in particular, especially that you did actually compare
Yeah,
smokers to non-smokers. Yeah, go ahead.
I think you are bringing up a very good question, and that's exactly what we wanted wanted to do, and we have been trying to do. But to do that, I think firstly, as you know, the location of adenoma and the squamous carcinoma is different, right? And at this moment, the biopsy we can collect it mostly from the bronchoscopy. That only goes to like the bifurcation of lung, but not all the way to the the what's that the lip. Sorry, I forgot the word. The 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 location of adenomas occurrence area. So that is the challenge for us at this moment. But we are trying to ask different lab which may harvest the sample from the adenomas occurring uh, area, I mean the normal cell there, so that we may get more insightful information or about the mutation pattern of the cell in the adenocarcinoma patients. At this moment, because most of our cells are harvested from the bronchoscopy, so we may not be, and plus there are in this study there are only 33 subjects. If we further divide them to subgroup, we may not have too much power,
Yeah, right.
statistic power to give like information. But even with these 33 subjects, we did analyze by subdivide them, and we did not really see. I mean, we did notice that adenocarcinoma patient, they generally smoke less, but they tend to have higher mutation frequency compared to squamous. But we don't really have statistic power at this moment to share this information in the publication.
Okay, that sounds great.
Do you have an estimate on how much more data you would need before you get enough power?
Of course, the more the better. We are challenged with either the cost of the sequencing, even though it has been reduced a lot, but still, for each cell, it was like $800. Yeah, so it is really very expensive. This is one thing. The other thing is because we are collaborating with a clinical lab. If So I don't think in the repository, the number is not like overflowing. So we have to collect more sample to make further study.
But I mean, towards Io's question, I think you could do a subgroup. I'm sorry, you could do a power calculation if you want to do a subgroup analysis and essentially try to get an idea of how many persons, how many subjects you'd need to recruit in order to, do that analysis in a way that would allow you to make a conclusion that would be statistically solid.
Yeah,
Yeah.
like we've done that kind of calculation ourselves with our sensor, and it was something like between 100 and 200,000 measurements had to be performed before an adequate calibration algorithm could be established. And we did that after a few hundred measurements. We did a few hundred measurements, looked at the analysis, and then worked back from there.
Okay. I think I know what you mean, but in reality, to make all these measurements, we need to balance between, I guess, the cost and also how much time
Logistics,
we need
sure.
to invest
Yeah.
into it. So so we are trying to maybe use the blood as the alternative. So that may help us to easily to get more sample with more subjects.
Oh, okay. So you would want to just look at like macro properties or some sort of correlation, or directly just look at blood measurements?
Yeah, first, you're right. We first need to establish or find the correlation, like from the blood or buccal swab, they say, because it will be much easier to get all these samples. Yeah, this is the first step, and that is what we are doing. So that will, and we will just exactly do what you have been saying, that calculate how much more sample we may need to have a static solid analysis to get a conclusion.
Thank you very
Yeah,
much,
we
Doctor.
found that
This
our
is going to give you...
calculation. Sorry, if I may just quickly, our calculation showed that it was cheaper to build a whole new kind of robot just for this task than it was to use human labor. So that's kind of a sad but funny kind of twist in our journey.
Yeah, this will just give you one more reason to come back sometime soon, Doctor. Thank you very much for your talk.
Yeah, thank you so much. And I just wanted to mention to the questions before, when you write a grant for NIH or so, they ask you to do that anyways, to do these calculations, what the power will be, how many like to basically excuse the costs that you will have for doing
Yeah,
stuff.
exactly. Yeah.
Oh, yeah.
So everyone does that, but what the problem is then what actually comes out of it. It's sometimes different, and we are discussing basically what the actual outcome was, not what we write on, in a model to get some grants. So, but yeah, I'm really looking forward to hear, because I had also ideas to screen for mental health and so on, for and check for markers of resilience in drawing blood and buccal swab. So I'm really curious about your next data, and also about the new technology you mentioned. So yeah, feel invited back. I hope you enjoyed being here with us today. And
yeah, thanks for all the great questions. I think it triggered me really a lot to think more about how to further proceed with this project. Thank you so much.
yeah,
If you don't mind, Professor, I'll probably be emailing you, and perhaps Katerina will be so kind as to share your email. And I think there is perhaps an opportunity to collaborate. I really like the idea of correlating to the blood. That is something that one of the teams here is working on, so that might be something that's possible. So
Yeah,
thank
sure.
you again for
Yeah, I would
that.
love to. Thank you so much for your interest.
wonderful. Another collaboration. I'm always happy to hear when people connect here.
So how could I share my email information?
I can connect you through email, if that would you.
Yeah, yeah.
Okay,
Yeah, that'd
I'll
be
write
great. Thank you
both
so
of
much.
you an email to introduce, and it's, you already were introduced, but just that you have each other contacts. And yeah, I'm always happy to hear when people connect here and start collaborations. It's not the first time, so I'm really glad. This
Thank
makes
you. Yeah, thank you
me.
for your organization.
Okay, yeah. So thank you everyone for asking wonderful questions, and thank you for sharing your wonderful and very important work you're doing. Again, I compliment you and congratulations. And yeah, talk with you. I hope I see everyone back, or hear everyone back soon. And yeah, stay tuned for more rooms. We have a room again tomorrow. And enjoy the rest of your day, evening, morning, wherever you are around the world. Bye.
Bye. Thank
Thank
you
you.
so much.
Bye bye.
Thank you.
Bye, Doctor.
Bye.
Bye,
Bye.
everybody.